Hello, everybody. Here we go with inheritance of multiple traits. Again, remember trait equal to, whoops, I erased it, gene. So studying single traits is fun, but not as fun as studying more than one trait. So Mendel finished up his work on single traits, and he wanted to know if the same rules applied to inheritance of multiple traits, two or more. So he looked at two or more traits. Let's look at his list of traits again. So we have seed shape and seed color, blah, 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 and down the line. So first he looked at this pair of traits, how they were inherited together, okay? So smooth, for seed shape, smooth is dominant over wrinkled, and yellow is dominant over green. Okay, so again, our allele symbols were assigned as follows. Pretty much just did that on the previous slide. So, whoops. When you cross between two true breeding varieties of each characteristic, okay, one dominant for both traits, the other recessive for both traits, we have two plants with these genotypes here, okay, dominant for both traits, this one, and this one is recessive for both traits, okay? This plant can only produce gametes that contain the dominant alleles, okay? And this plant can only produce gametes that contain the recessive alleles, so our Punnett square is actually pretty boring. It's only one box. When you make your zygotes and we reunite the gametes, you're going to get all the offspring from this cross is going to be heterozygous for both genes. Okay, so the F1 generation is heterozygous for both genes. Okay, don't pay any attention to this because it was too long the first time and I had to go through and redo it. So let's get all this stuff off of here. Missed a slide. Okay. So Mendel next did what he normally does, take the F1 individuals and allowed them to self-fertilize. So he crossed two plants that were heterozygous for both genes, and we call this a dihybrid cross. Okay. This cross yielded offspring that were just like the parent plant with dominant for both traits, about 315 of those out of 500. They yielded offspring that were recessive for both traits, about 32, wrinkled and green. And then what was interesting is that they yielded some recombinant plants that had one recessive and one dominant trait. Okay, about 101 of those out of 500, and about 108 of the other combination, dominant and recessive. Okay, and this gave us a phenotypic ratio of 9 to 3 to 3 to 1. And every time he crossed two genes, whether he looked at tall and flower color, or seed pod shape and pod color, or seed shape and flower color, it didn't matter what pair he looked at, they all came up with this phenotypic ratio. So this is something you just need to learn because it's too complicated to figure it out. So the dihybrid cross phenotypic ratio is 9 to 3 to 3 to 1. Let's look at if you want to do out the Punnett square, this is what it's going to look like. Okay. So here are my gametes that can be produced by this plant. Okay. Remember, we're going to segregate the alleles, so separate the alleles. And we're also going to combine the alleles with each allele from the other gene. So this dominant allele can be in a gamete with this gene, or it can be in a gamete with this allele. This, the recessive allele, can be in a gamete with the dominant allele here, or the recessive allele. And that gets you your following gametes here. You can have this combination or this combination. and this combination, or that combination. Okay, once you put them in a Punnett square, put everybody together, you get your 9 of the double, there's my 9 of the double dominant. You get 3 with 1 dominant and 1 recessive. You get another 3, 1, 2, 3, with the other dominant and the other recessive, and you get 1 with the double recessive. Okay. Now, what does this all mean? So Mendel hypothesized that traits are inherited independently. And if this is starting to sound like the law of independent assortment, you are right. If it wasn't starting to sound that way, guess what? It's the law of independent assortment. So if the two traits were inherited independently, then each trait by itself should show that 3-to-1 ratio that he had seen for a single trait. And that's exactly what they did. So he had 423 plants, had smooth seeds, which was the dominant trait, and 133 plants that had wrinkle seeds, that was the recessive trait, and that's about a ratio of 3-to-1. Same exact thing for looking at the other trait by itself, 416 yellow, which was dominant, 140 green, which is recessive, again, giving us about a 3-to-1. So it seems like these genes aren't really affecting each other. So they're moving independently. And this is where he came up with his law of independent assortment. And it was figured out later, as we already know, that this is because of how the chromosomes, the homologous pairs, line up independently of each other at the metaphase plate in meiosis 1. Okay? Multiple traits are inherited independently because the alleles of one gene are distributed to gametes independently of alleles of other genes. Okay? And the genes have to be on different pairs of homologous chromosomes. We'll talk about what happens when they're on the same pairs a little bit later in the chapter. Okay? And again, physical basis of the law of independent assortment has to do with meiosis 1, metaphase 1 of meiosis 1. Okay, whichever the homologs is on top assorts randomly, and the other assorts randomly and independently, and then they separate in anaphase 1. So here's a nice graphic of that, and you can see here can line up in 2 different orientations, okay, independent of each other. And so this step here is the law of independent assortment. And then later on in meiosis 2, when we separate the sister chromatids, we have our law of segregation. And you just get 1 copy of each allele in each gamete. Okay, and down here we have all of our possible gamete combinations. And that's how you get it, the gamete combinations for the dihybrid cross, or for any cross. Okay, so this is a pretty good graphic to know. It demonstrates both laws right there and their relationship to meiosis. And I think that is all done with Mendelian genetics. Okay. Tune in to the next podcast where we talk about how there are exceptions to every one of these rules that I've just laid out for you. You're welcome.